Good morning, everyone. My name is McKayla Nicol. I'm a senior scientist at BioModels. Thank you all for joining me today for this webinar. As I'm sure you are all aware, the purpose of this presentation is to review neuro-behavioral testing in the context of preclinical models. To give a brief outline for the time that we have today, I'll start by giving a quick background on biomodels. I'll review a variety of behavioral tests, and then I will provide some example studies where these tests were implemented before closing out the webinar. And I just wanted to note that we will have the Q&A function open for the duration of the webinar, so please feel free to submit any questions that you might have throughout the talk, and we'll follow up with an email after the fact. So to start, BioModels is a preclinical contract research organization. We are located in Waltham, Massachusetts, right outside of Boston. We were originally founded in 1997 out of the Brigham and Women's Hospital with a handful of animal models, and we've since expanded and now offer a variety of highly translational models of human disease in rodents. We serve a really diverse client base, and we're always welcoming the opportunity to collaborate with anyone who has needs in the preclinical space. Just a few of our areas of expertise include inflammation and autoimmune diseases, fibrosis, pulmonary diseases, microbiome manipulation and treatment studies, oncology, cancer supportive care, and we're also always open to working with our clients to create custom models that are specifically designed to best support their unique program. And since the origination of the company, we have helped our clients bring over 100 compounds to the clinical space across a variety of the indications that we model. As I already mentioned, the purpose of this presentation is to focus on behavioral assessments and discuss why they're important for preclinical models. These tests can really provide insights that are going to be complementary to biological data and histopathology analyses. And when all of the data are combined, that helps to offer a more comprehensive understanding of disease severity and potential therapeutic impact. And here at BioModels, we have the capabilities to support testing across a really wide range of categories, including depression, anxiety, pain, neuropathy, motor dysfunction, and many others. And I'd also like to note that behavioral assessments can be incorporated into any kind of preclinical models. And so they're not limited to classical neurodegenerative or neuromuscular disease models. And so these tests can be really flexible and allow you to select the assays that are going to provide answers that you need for your study objectives. Moving on now to the first information segment of the webinar, I'll be starting with the review of some of the behavioral assays. This table is a general overview of some of the tests that BioModels has run in the past and that we continue to run. We've categorized them into groups of depressive-like behavior, anxiety and fear-related testing, cognitive testing and memory assessments, motor coordination, social behaviors and interactions, and circadian rhythm and sleep behavior. And while this is not a fully comprehensive list of all the tests that we have done or that we have the capabilities to perform, it does give a good general overview with examples of tests in multiple categories. Though that being said, any one of these tests does not necessarily fall into only one category and could potentially be classified in different ways depending on the model that it's being tested in. And so keeping that in mind, I'll go into each one of the tests listed here with a little bit more detail to give a better idea of the methods and applications for the assays. Starting off with the forced swim test, which is also sometimes called the Persault swim test. This was actually first developed for rats and then later modified for mice. For this test, animals are placed into a cylindrical chamber with water and monitored for their behavior. It's a pretty common test that evaluates endurance and exercise-induced fatigue and is primarily used to measure depressive-like behavior. So the test subjects will eventually stop trying to escape and they'll exhibit what's described as behavioral despair. And so for the readouts of the assay, the researcher has to distinguish between what's called the active coping stage, when the animal would be swimming and climbing and continually struggling to escape, And then the passive coping stage where the animal is much less mobile, mostly floating and generally using as little movement as possible just to remain above the water. The tail suspension test is similar in concept to the force swim test, wherein escape-oriented behaviors are assessed. But here the animals are suspended by their tail instead of being placed into water. And this does remove the risk of hypothermia that can be associated with the water submersion protocol. But there are also issues associated with the tail hang test. One of the major limitations is that animals can learn tail climbing behavior, and animals that successfully climb their tails then learn that escape is possible, which effectively negates the premise of the test. But despite potential issues for either of the tests, both can be used to screen antidepressant compounds, where in theory the compounds could extend the time that the animals would spend in the active coping stage. Next, for the fear and anxiety category, we have included open field, elevated plus maze, and self-grooming assessments. For open field testing, animals are placed into an empty enclosed chamber, given some acclimation time, and then subjected to an observation period. And this test is a really popular platform in the realm of behavioral testing, and it can be used to measure animal behavior, providing information on general ambulatory abilities as well as the emotionality of the subject animal. And general ambulation is usually measured by the overall distance traveled during the observation period for the test. And then the emotionality or anxiety related assessment can be measured because rodents have a natural aversion to brightly lit or open and unknown environments. And so animals with higher than normal anxiety might have a tendency to stay closer to the walls of the chamber and avoid the open and brightly lit center region, even more so than a normal healthy animal. The number of fecal boli left during the observation period can also be a measure of anxiety for this test. And then the elevated plus maze does similarly rely on rodents' aversion to open and, in this case, elevated areas. For this test, a plus-shaped maze is set up with 2 open arms and 2 closed arms, and animals are placed into the maze and observed, and the number of entries and time spent in each of the arms of the maze would then be recorded. And then lastly in this grouping is the self-grooming assessment. Grooming behaviors are typically considered a maintenance behavior for rodents, but self-grooming can have other implications, including self-soothing for pain or anxiety, which is why this assessment is included here. Placing animals into a chamber and monitoring their grooming habits can reveal anxiety. It can also be a measure for certain behaviors in disease models like autism, to look at measures of repeated behaviors. Cephalocaudal grooming specifically is a pattern of self-grooming that is outlined based on behaviors of healthy animals. And so rather than just assessing number or duration of grooming events, researchers could also use this pattern to detect disruptions or highlight different steps that could be performed in excess that can happen with animals subject to chronic stress, or in some transgenic animals like Hawksby-8 mice, which use these mice are used to model trichotillomania. But overall, all 3 of these tests can be used to screen anxiolytic or anxiogenic compounds, in addition to being used for alternative disease-specific indications. Moving on to testing for cognitive thinking and memory, 2 of the commonly used assessments in this area are the object recognition and fear conditioning tests. The object recognition test is used to look at the memory of animals by exposing them to different objects and then observing their interactions. General expectations for a typical rodent would be for them to spend more time investigating new objects over those that they have already seen. And so if exploration of new and old objects were to be the same in this test, a researcher could potentially interpret that as a deficit in memory or learning. And this test is also very versatile. And so by moving objects or changing objects, different phases of learning can be assessed. The test can also measure different regions of the brain and how different therapeutics or dietary supplements might impact function. And so, because this assay can be used to look at modulation of cognitive function by a number of factors, it's popular for use with Alzheimer's models and also aging and development studies. The fear conditioning assay also measures associative learning and memory, but through recollection of traumatic events. Contextual and cued fear conditioning are the most commonly used methods here. Contextual conditioning involves placing the subject animal into a new environment where an aversive stimulus is introduced. The animal would then be removed and then after a period of time placed back into that same environment. And typical behavior would be a freezing response, indicating that the animal remembers the environment and is associating it with that aversive stimulus. Cued conditioning presents the animal with a cue, such as a flashing light or an auditory tone, followed by the aversive stimulus. And after the training period, the animal is again expected to demonstrate the same freezing behavior in response to the cue, as it has theoretically learned that that is an indication for the subsequent aversive stimuli. In addition to general memory assessments, the fear conditioning tests are often used for PTSD models to investigate neural circuits involved in processes of developing fear and fearful behaviors, and then therapeutics or treatments for potential reversal. The next category that we have are the motor skills and coordination tests. And here we're going to talk about rotarod, forced running, and grip strength tests. Rotarod is one of the oldest tests used to assess neuromuscular coordination in rodents. For the test, animals are usually placed onto a motorized rod that can be set to turn either at a constant or increasing speed, and then the time that it takes for the animals to fall off of the rod is recorded. And this test is often used to assess basic motor deficits in neurodegenerative diseases like Parkinson's, Huntington's, or multiple sclerosis. The forced running test is used to look at exercise endurance in rodents. The test uses a motorized treadmill. Animals are placed onto the belt, and when they stop running, they slide back onto a grid at the back of the treadmill belt that can deliver an electric shock, which would then prompt the animals to return to running. This is typically done for a set period of time, and the researcher would be responsible for assessing the animals to see if they've reached a point of exhaustion and need to be removed from the test. Forced running can also be used for a couple of different applications. It can be introduced for muscular dystrophy or other neurodegenerative models to exacerbate disease progression. It can also be used to investigate the effects of exercise as a treatment. It can use to measure fatigue or endurance in any number of metabolic disorder models as well. But the forced running test can be quite stressful for the subject animals, so that should be a consideration when planning for experiments that may include it, and other stressful testing should be limited within the battery. And then finally, grip strength testing is used to measure forelimb strength. Animals are going to be suspended by their tail for this test. That causes them to instinctively grab for the bar or grid of the grip strength meter. The researcher would then gently pull back on the animal until the bar is released. And this assay can be informative for muscle physiology in really any of the previously mentioned neuromuscular diseases, as well as potentially in aging studies. Moving on to the social behavior and interaction category, the primary test that we use at BioModels in this category is the social approach or three-chamber sociability assay. This test quantifies direct social behavior, and it's based on rodents' natural tendencies to be interested in new or stranger animals. There are, again, several variations of this test, but generally animals are going to be placed into a box that has three chambers. They are allowed to acclimate, and then one or more stranger animals would be introduced in contained holders into the chambers of the box. And then the amount of time that the test subject would spend investigating the stranger animals or empty containers is analyzed. This test was originally designed for investigating sociability defects in mouse models of autism, but it has since been adapted to other neuropsychiatric disorder models like schizophrenia, and depression. And now our last category for the behavioral assays is general behavior, sleep, and function. And under this grouping, we have wheel running and light dark reversal. So wheel running here is referring to voluntary wheel running, which is quite different than the previous forced running that we reviewed. Although it can also be used to assess physical performance and endurance or to model exercise training. But importantly, this is done without any adversive stimuli, and so this test can be used to assess metabolic health or general mental health, and also has potential for use with chronic pain models. And because there is no stimuli, it's not generally considered to be stressful for the animals and is done spontaneously according to the natural rhythmicity of the animal. And then light dark reversal is something that can be applied actually pretty much for all behavioral studies. Because rodents are naturally nocturnal animals, there is a marked improvement in performance during peak activity hours. And so it can be helpful to artificially reverse their cycle, and enable researchers to perform these tests during daylight hours. But also changes in light dark cycles could be used to research circadian rhythm disruptions and how that impacts mood and behavior. So this capability could be used for multiple different purposes in behavioral research. So that brings us to the end of the list that we originally outlined in that table to review here, with tests that are routinely performed at BioModels. But of course, there are many, many other behavioral assays that can be used for preclinical research studies, some of which are listed here. But again, this is also not by any means an exhaustive list, more just a sampling to give an idea of the magnitude of this space. And of course, this also then doesn't include additional variations that can be introduced to each test that can tailor them even more specifically to a particular need or program or area of interest. Building off of that, though, it's easy to see why it can seem like there are an overwhelming number of options to choose from when selecting your behavioral battery. But I've simplified the process here with a few considerations to keep in mind when going through steps to design an experiment. Generally, to start, you will, of course, need to identify your research goal. Then once you have the objective and hypothesis defined, you'll need to select the model that you want to use to assess the phenotypes of interest, whether those be disease-related or in healthy animals. You'll want to set your experimental conditions, so selecting appropriate group numbers to ensure a sufficiently statistically powered study, adding correct control groups for comparison, selecting treatment doses and timelines. Once those parameters have been set, you'll then want to select functional tests that will assess the target behavior in a sensitive and consistent manner. Something to keep in mind is that it's usually recommended to have at least two overlapping tests so that you can have multiple measures of the same construct within a study. Other factors to consider here are going to be practical constraints, so things like time, expertise, software, equipment. The order that the tests are performed in will also be important for animal welfare and data consistency. And you should also definitely consider the general repeatability and reliability of the tests. And a relevant metric for those factors would also be how well published the assays are. And then, of course, you'll want to make sure that the behavioral battery data can act as sufficiently translational correlates for the ultimate goals for the clinical outcomes. And you'll then want to assess external considerations such as the experimenter interaction with the subject animals, conditions of housing, the testing areas. And then finally, you can run your experiment and analyze the data. So now that we have reviewed some behavioral test options and talked about how to choose your battery of tests, I'm going to walk through some examples of studies that went through the process and share some sample data, both behavioral and biological, to give you a better idea of how you can incorporate these assays into preclinical test models. The first example that I have for you is a study for autism spectrum disorder, or ASD. ASD is a neurological and developmental condition that typically presents in early childhood. It's characterized by substantial abnormalities in functional brain connectivity. These differences in brain connectivity result in a pretty wide range of symptoms and symptomatic severity. And the most defined symptoms for the disorder include differences in sociability, repetitive patterns of behavior, and sensory sensitivities. Current treatment options for ASD are largely targeted towards symptom management through behavioral, speech, or occupational therapy, or a combination thereof. But there is a continued need for research to help us better understand the roles of genetics for the etiology of ASD and hopefully in turn improve early diagnostics so that the therapeutic behavioral interventions can provide really as much support as possible during early developmental phases. Moving into animal models and specifically mouse models that replicate ASD for preclinical research, there are really a plethora of mouse models that have been genetically engineered to produce translational behavioral phenotypes. In a recent publication by Zerbe et al., I have a figure here that showed brain-wide connectivity mapping that was done across 16 of the more popular mouse models of ASD. The research group was able to classify the 16 models into 4 subtypes based on the altered connectivity signatures. And based on what's been shown in other publications, the behavioral phenotypes of the models are not necessarily simplified into the same cluster pattern, but this data does show clinically relevant variations in the etiology of ASD, which result in heterogeneous functional brain connectivity and a range of symptoms and symptomatic severity, which again is of great importance for ASD diagnostic research. And so focusing in now on a singular mouse model, we have chosen to use the BTBR mouse model of ASD, which was included in cluster 2 of the brain mapping project. The BTBR model is a very highly published and well-accepted translational model for ASD. This slide shows an example study design outlining the parameters of group numbers and some behavioral endpoints just to assess the basic phenotypic standards within the model. Normal readouts for this would include weight monitoring, the 3-chamber sociability, and then a measure of repetitive behavior. So coming back now to the social approach assay that I mentioned before, just to review the methods for the test, we would typically place the animal into the enclosure, allow them to acclimate for 5 minutes, and then a stranger mouse and an empty cup would be added into opposite chambers, and then those cups would be swapped for an additional 5 minutes. And so this is the resulting data from the 3-chamber assay run with BTBR transgenic mice and wild-type mice. The animals did travel similar distances throughout the duration of the experimental phases of the test, as seen in the graph on the left. And the animals spent a similar amount of time investigating the stranger mouse, as shown in the blue bars in the graph on the right. But importantly, the wild-type mice spent significantly less time investigating the empty cup relative to the time spent investigating the stranger mouse, which was not the case for the BTBR animals, and that's what is indicative of the reduced social discrimination for the transgenic mice. We also tested rotarod and self-grooming in this model. For rotarod tests, we do a training session with the animals prior to the data collections. On the day of the assessment, then we would run a series of 4 trials on the rotarod, which would be set to a constant speed, and then the average time to fall for each animal is recorded. For the self-grooming, we place the animals into an enclosure and allow them to acclimate and then observe and record any grooming behaviors. And so data from these assessments, again, reflect differences between the BTBR mice and their wild-type controls. The rotarod data on the far left, while not significant, does show a notable decrease in the average time to fall for the BTBR mice relative to the wild-type controls. This difference in the ability to remain on the rotarod is indicative of abnormal cerebellar development, which is linked to motor coordination and learning. This abnormal brain development can also lead to dystonia-like hyperflexion, which is another known BTBR phenotype and does impact the animal's ability to remain on the rod. Next, the center graph shows the number of grooming events, and the right graph shows the duration of grooming, both of which are significantly increased in BTBR animals relative to the wild-type animals, which indicates a clear increase in the repetitive behaviors associated with grooming and potentially also increased levels of anxiety in the BTBR mice. So those were just a few behavioral tests that demonstrated the phenotype of the BTBR animals. There are, of course, other behavioral tests that could be incorporated into ASD studies, and those could include options like open field or an elevated maze system to assess anxiety, or additional repetitive behavior assessments like the marble burrowing or whole board test could also be used here. Moving on from ADS, the next model that I'm going to review is for Duchenne muscular dystrophy, or DMD. Muscular dystrophies are another class of genetic disorders that are generally characterized by loss of muscle tissue and subsequent function. Duchenne muscular dystrophy is an X-linked recessive disorder caused by mutations in the DMD gene that results in a lack of dystrophin protein. Clinical signs of DMD typically start pretty early with delayed walking and early indications of muscle damage. By the mid stages of disease progression, most individuals have loss of ambulation, and then typically by early 20s, there's significant respiratory and cardiac dysfunction, which leads to mortality. Previous treatments were largely focused on prolonging the period of ambulation and delaying that onset of more severe and eventually terminal symptoms. But there are actually some newer approved gene therapies with ongoing research in this area for targeting curative levels of treatment for the disorder. Narrowing down on mouse models of DMD, these are three of the most commonly used mouse models, each with their own strengths as translational models of disease. The model at the top of the figure are B10MDX mice, and they do exhibit similar pathology of the diaphragm to what is seen in human DMD, but the muscle degeneration and lifespan of the animals have a more reserved phenotype than what's seen in human disease. The center model are DKO, or double knockout mice, and they are double knockouts because they are lacking both the dystrophin and utrophin genes. And so this creates a different genetic signature from the human disease because of the additional impact to utrophin. But the resulting disease pathology of the model is more similar to what is seen in humans, with progressive limb degeneration and premature death, in addition to the respiratory damage to the diaphragm. Lastly, the bottom model is the most recently developed of the three and is an MDX mouse on a DBA background as opposed to a C57. And this model is sort of a good compromise between the other two because the pathophysiology is more severe than what's seen in the C57 MDX model, and the genetic mutation doesn't impact the utrophin gene like it does in the DKO model. But one downside to this model is that it can present with calcifications in the muscle, which is something that we don't see in the disease in humans. So you have to control for that appropriately with vehicle and naive groups in these studies. So at BioModels, we have historically used the D2-MDX mice to model DMD. This again is a sample study design with some planned behavioral testing and then terminal collections for histopath analyses to confirm the muscle pathologies. This is some of the histopath data from the diaphragm and quadriceps muscles of D2-MDX mice compared to wild-type DBA controls. The samples from the transgenic animals are shown on the bottom for both slides here, and you can see that there is clear evidence of lesions of muscular dystrophy in the MDX mice compared to the controls. Both tissues are showing myocytes with areas of necrosis and mineralization. There are areas of inflammation and regeneration also visible in the samples of the diseased animals, and this is all pretty much exactly what we would expect to see in animals for this disease indication. And so for the behavioral testing aspects, we again use Rotarod with the same protocol that I previously described. We also incorporated open field testing to assess general ambulation. And for the open field test, the animals are placed in the chamber, allowed to acclimate, and then observed and recorded. And so the graph on the left here is showing the Rotarod data and pretty clearly demonstrates that the control animals have a longer average time to fall from the Rotarod over the course of the study. And the area under the curve was analyzed, and the overall difference was found to be statistically significant as shown by the graph in the figure inset. And the open field distance parameter, which is graphed on the right, does indicate that the control animals were notably more active within the chamber, although that readout was not significant by the same AUC analyses. We also looked at grip strength and hang test assays for these animals. For the grip strength, we had a pre-study training session, and then for each time point during the study, the animals are subject to a series of 5 trials being pulled off of the bar to measure average pounds of force applied by the animal before releasing the bar. The hang test also had a pre-study training session, and for this test, animals are placed onto a wire mesh that is then inverted, and the time that the animals hold on before dropping off is recorded. For the grip strength testing, which is shown on the left, again, the wild type animals demonstrated significantly higher pound force averages across the board compared to the D2-MDX mice, and that indicates decreased muscle strength in the diseased animals. The D2-MDX mice also did demonstrate a lower average time to fall from the wire hang test, as shown on the right, which means that they were not able to hold themselves up as long, although that difference was not statistically significant. Some additional testing that could have been incorporated for this model could include forced treadmill running. It's not entirely necessary for the D2-MDX mice as they do spontaneously develop progressive muscular decline. However, for the C57-MDX mice, forced treadmill is sometimes included to exacerbate the disease phenotype. Wheel running is another test that could be used to assess fatigue and exercise attempts with these animals, and then balance beam testing could be used to assess motor coordination as well. So the 2 models that we just went through were both transgenic models of disease that benefited from behavioral assay readouts to evaluate disease severity or model phenotypes. The next 2 models that I'm going to be going through are chemically induced disease models, so no genetic modeling, but they will also utilize behavioral assays. And first, we are going to look at Parkinson's disease, or PD, which again is a pretty classic neurodegenerative disorder. It is characterized by the loss of dopamine-producing neurons and results in slowed movements, tremors, loss of muscle control, as well as a variety of non-motor symptoms. Current treatments for this disease focus on largely symptom management, but research does continue to branch out with focuses on stem cell transplants and other disease-modifying therapies with potential to replace dopamine cells rather than just mitigate symptoms. Although other models do exist, as I mentioned, we're going to focus on chemical inductions here. So some induction paradigms used to model Parkinson's in rodents include MPTP, 6-OHDA, rotenone, and paraquat. These neurotoxic models of PD have been found to be highly reproducible, and they're popular because they are able to rapidly and pretty reliably induce disease phenotypes. MPTP is the most commonly used model. It can be easily injected since it crosses the blood-brain barrier, and it causes selective destruction of dopaminergic neurons by targeting the substantia nigra, pars compacta, and striatum. 6-hydroxydopamine is more commonly used in rats, and it does also selectively destroy the dopaminergic neurons, but it does so by disrupting mitochondrial function. 6-OHDA cannot cross the blood-brain barrier, though, so it does have to be directly injected into the brain. Rotenone and paraquat are other neurotoxins that are used less frequently, but rotenone is a naturally occurring neurotoxin and paraquat is a herbicide, and both can cross the blood-brain barrier and cause selective destruction of the dopaminergic neurons. At BioModels, we have largely used the MPTP model of PD. The table on the left is again showing a sample study design that includes a treatment paradigm and rotarod testing with brain collections for histopathology. This is some sample histopath data of CNS samples showing the loss of the dopaminergic neurons in the MPTP model with the diseased animal sample on the right and the healthy comparator on the left. Tyrosine hydroxylase was used to stain the substantia nigra, which is what is specifically targeted by the neurotoxin in this model. The disease pathology was also monitored with rotarod, where mean time to fall was notably greater for the MPTP treated animals relative to those administered a vehicle control. And this is an expected outcome as the release of dopamine into the striatum is essential for both the promotion and inhibition of movement. So the loss of the neurons responsible for regulating the nigrostriatal pathway is going to have significant impact on motor activity and control. And then here again, we have some additional options for different behavioral testing that could be performed with the PD model. Forced treadmill is an option to look at motor coordination with potential for disease exacerbation. The pull test is one that can be performed to specifically assess bradykinesia or rigidity, which is a hallmark symptom of PD directly caused by dopamine deficiencies in the basal ganglia. Open field testing can also be done for anxiety and general ambulation, and then grip strength tests to look at muscle function and strength. Moving on to the second chemical induction model for today, we're going to go over cancer-related fatigue. Fatigue is actually the most common symptom in both adults and children with cancer. Fatigue occurs at different levels in the broad population of cancer patients, but specifically for those receiving chemotherapy treatments, over 80% of those patients report fatigue as a side effect. And current options for this can include medication and lifestyle management, but research is still ongoing to try to identify risk factors for the development of the more severe and lasting cases of fatigue, as it can actually last for years even after treatment has completed, which obviously can be extremely distressing and a very significant detriment to quality of life for these patients. For mouse models of cancer-related fatigue, a variety of chemotherapy regimens, radiation, and tumors have all been used alone and in combination to induce the fatigue phenotype. At BioModels, we do have extensive experience with both chemotherapy and radiation-induced fatigue models. The data that I have here is from a chemotherapy-related fatigue model using doxorubicin as the induction agent. And so sample study design, including a therapeutic treatment and then use of wheel running as a measure of fatigue for these animals. For this study, the animals were singly housed. Wheels are added to the cage after treatment, and animals are allowed to run on the wheels as they please. The number of rotations of the wheels is recorded and translated into distance per 24-hour period throughout the study. And the graph on the right there clearly shows that the animals administered doxorubicin demonstrated much lower levels of spontaneous activity relative to animals that are left untreated or that were given saline as a control. Some additional behavioral testing that could be done here includes rotarod to further assess fatigue and motor skills, open field for general activity and anxiety, treadmill running for fatigue, and forced swim for measures of fatigue and depressive-like behavior. All right, so now that we have gone through some of the more classic models of neurodegenerative and behavior-related disorders, both transgenic and chemically induced, we are going to pivot to one final example of a disease model that benefits from the inclusion of behavioral testing. But the use of behavioral testing in this case could potentially be considered slightly more tangential to the typical expected use of these assays. Though that being said, it's still a good addition to the study and provides useful and informative data for the model. And that model is atopic dermatitis. Atopic dermatitis or eczema is a condition that causes dry and itchy inflamed skin. It's a chronic condition that can occur anywhere on the body, and it can persist even with topical medications. Research for the disease indication is also still ongoing, and newer studies have been done using therapies with microbiome restoring topicals or personalized targeted treatments for individual patients. And then there are lots of mouse models for atopic dermatitis, both induced and spontaneous models. At BioModels, we do run several of these models of dermatitis, as well as others that are not listed here. For the purpose of this presentation, we'll be focusing on the induced models, of which there are still multiple categories here, including bacterial, diet, chemical, and allergen inductions. Specifically today, we're going to be looking at a house dust mite or HDM induction, and so an allergen-based model of atopic dermatitis. Here, the allergen was administered in conjunction with Staph aureus and enterotoxin B, which acts as an antigen adjuvant that enhances the inflammation for the model. We monitor body weight, perform clinical scoring, and do histopathology for these animals. And the behavioral assay that was introduced here is a scratch test. So first, going through the histopathology results, representative figures are shown on the right, with the healthy animal sample on top and the diseased sample on the bottom. You can see pretty clear evidence of epidermal hyperplasia, dermal mixed inflammation, and dermal fibrosis in the diseased animals, which is pretty much exactly what we would expect to see in a model of atopic dermatitis. And then here we're showing the biological data points of the percent weight change on the left and clinical scoring on the right, which includes measures of erythema, edema, excoriation, dryness, scarring, and lichenification. And the HDM subgroup had similar weight trends, but significantly increased composite scores compared to the healthy animals, indicating successful disease induction. Then for the scratch test, we placed animals into a chamber, allowed them to acclimate, and then recorded activity to assess any scratching behaviors. And the difference between the start of the study compared to the end of the study is pretty clear with the HDM sub animals scratching much more frequently and for extended durations in the observation window compared to the vehicle control animals on day 29. And again, although this is not a traditional neurodegenerative disease model, there are plenty of other behavioral assays that could be incorporated into the study design to collect a more comprehensive data set on the disease phenotype and animal state. For example, the von Frey filament test could be used to assess mechanical allodynia or sensory dysfunction in the inflamed regions of skin. Similar assessments for the thermal sensitivity testing. And then these animals have also been reported to exhibit some higher levels of anxiety. And so assessing them with open field or light dark box tests could also be valid options here. So that concludes the review of example disease models with behavioral testing. To close out the webinar, I first wanted to give a quick summary of what we've gone over with respect to neurobehavioral testing. As I mentioned earlier, there are, without question, many options for neurobehavioral tests that can be applied to preclinical models, both in the traditional neuromuscular psychiatric behavioral space as well as outside of that. And while there are a lot of factors to consider while choosing the behavioral battery that would be the right fit for your preclinical program, there's plenty of resources available. And here at BioModels specifically, we have the capabilities and expertise to provide you with whatever support you might need in this area. Coming back to BioModels as a whole, in addition to the neurobehavioral space, I'd also like to reiterate that we do work in a variety of other disease areas as outlined here. And even if you don't see what you're looking for on this slide, we are always happy to discuss custom modeling as well. And as I'm sure you can gather from the number of different models that we offer, BioModels has pretty extensive capabilities with different systems for in-life monitoring options for surgical models. And we also have a full in vitro lab and microbiology suite to support studies with in vitro analyses or microbiology components. And getting a project started with BioModels is very straightforward. You can contact us via email or on our website, both of which will be provided on the next slide. As you can see here, though, the process breaks down into 6 simple steps from initial contact to delivery of the final report. We do offer a comprehensive start to finish solution, and we hope to have the opportunity to collaborate with everyone who has joined the webinar today. And so that concludes my presentation. Thank you again to everyone who joined and stayed for this talk. We appreciate you taking the time out of your day to hopefully learn something from the webinar. And please feel free to reach out to us with any additional questions or to discuss potential projects.